Photosynthesis is the process by which plants convert light energy into chemical energy, producing glucose and oxygen from carbon dioxide and water. In this experiment, known as the Photosynthesis in Leaf Disc Lab, we investigate how light affects the rate of photosynthesis by measuring oxygen production in leaf tissue. The main idea behind this lab is that oxygen, a byproduct of photosynthesis, causes leaf discs to float. By measuring how quickly the discs rise, we can estimate the rate of photosynthesis under different conditions. The purpose of this experiment is to determine how light exposure influences the rate of photosynthesis in plant cells. Our hypothesis is that we believe that we can see that if the light discs are exposed to light, then they will float faster because photosynthesis will occur at a higher rate, producing more oxygen. In contrast, discs kept in the dark will float more slowly or not at all due to the absence of light. The materials that are used in this lab include fresh spinach leaves, a hole punch, a syringe without a needle, sodium bicarbonate solution, soap solution, cups that are 10 ounces, a light source, a scale with 0.01 gram precision, a timer, paper towels, a permanent marker, and a ruler. Now for safety precautions. Sodium bicarbonate is slightly toxic by ingestion. Do not consume. Remember, wear goggles when glassware is in use. Keep water away from electrical cords and outlets, and follow all other normal safety precautions as well. First, several leaf discs are punched from spinach leaves. Try avoiding major veins. These discs are placed into an empty syringe, then it is filled with sodium bicarbonate solution and a drop of soap. The air inside these discs is removed by pulling the syringe plunger to create a vacuum. This causes the discs to sink once the vacuum is released. Next, the discs are transferred into clear cups containing fresh bicarbonate solution. One cup is placed under a light source, while the other cup is covered up with a paper towel to block light. As photosynthesis occurs, oxygen accumulates inside the leaf discs, causing them to become buoyant and float to the surface. The number of discs floating is recorded at 1-minute intervals. Make sure to record the time it takes for 50% of the discs to float. Typically, discs exposed to light rise more quickly than those kept in the dark. The results should show that leaf discs in the light floated significantly faster than those in the dark. This indicates a higher rate of photosynthesis when light energy is available. The discs kept in the darkness should have shown little to no movement, confirming that light is required for the light-dependent reactions of photosynthesis. In conclusion, this lab demonstrates that light intensity directly affects the rate of photosynthesis. The floating disk method provides a simple but effective way to measure the oxygen production in plant cells. This experiment supports the hypothesis that photosynthesis occurs more rapidly in the presence of light and highlights the importance of light-dependent reactions in energy conversion.